Bismillahirrahmanirrahim. Assalamu alaikum. Welcome to my YouTube channel, Science for Everyone. Today, I have brought a very important video for you people, and this is mostly related to the students that are of microbiology. And in this video, I will solve different MCQs, and I have chosen these MCQs from very reliable and authentic books. And these are the MCQs that mostly come in competitive exams. For example, when a student is applying for MPhil in microbiology or biotechnology, so these type of MCQs come in their exams just to apply for the MPhil program. These MCQs are also included in different other exams like subject specialist exams and many other commission type exams. Okay, so let's start with the video, and I want to tell you guys that this will contain consist of two parts. Okay, in the first part I will solve 50 MCQs, and in the second part I will solve about 45 MCQs. So the total MCQs are 95, and I will explain those MCQs with explanations, with diagrams, okay, so that you are clear about it. So it will help you a lot. So please watch the video till end, and make sure that you should solve all the MCQs, okay. I want to tell you that I will give you a 5-second time, so just pause the video and first of all solve the MCQs by yourself. And if you are not able to solve it, so then I will show you the correct answer. Okay. So let's start with the video. Okay. These are the MCQs I have taken from a book, and some MCQs are very much easy, some are a bit technical and conceptual, so you will enjoy it a lot. Okay, the first MCQs is that which of the following statement help to explain how bacteria can become antibiotic resistant. Okay, it is about the resistance of an antibiotic, the resistivity that is established by the bacteria. So this includes that how will it get resistant. So the first is that the gene can produce protein that provide resistance. So I want to tell you guys that when there is an antibiotic against a bacteria, so that bacteria can produce genes that encodes protein that create resistance to the bacteria. So the A is correct. Now just look here to the second one. Spontaneous mutation can provide variation in gene function. Okay, so when there is antibiotic applied for a bacteria, so there can be spontaneous mutation just to overcome the just to fight against the bacteria, the antibiotic. Okay, so there can be genes can also be produced just to fight the antibiotic, and spontaneous mutation can also occur in its DNA. Okay, so both are correct. And the third one is that antibiotic use will select for bacteria that are resistant, that we all know. So the D option is correct. Okay, the D option is correct. Now the second one is the cell division in bacteria takes place mainly by, so here is conjugation, sporulation, fragmentation, binary fission, and we all know that bacteria can only mostly it can divide through binary fission. So the correct answer is B. Okay. Now the third one is that some bacteria form a thick-walled dish in response to poor nutrient condition. So we all know that when there is adverse condition for a bacteria, so it can then transform into another shape, and that is known as endospores. Okay, so the correct answer is endospore. Now fourth one is that the gram-negative bacteria do not stain purple as gram-positive do, because they have. So now this MCQ is about the differentiation between the gram-negative bacteria and the gram-positive bacteria. So just look here, gram-negative bacteria do not stain purple. We all know that it's stain pink. Okay, so why as gram-positive do? Because they have too much they have too much, wait a minute. Okay, Okay, so gram-negative bacteria do not stand purple because it is having less peptidoglycan. Okay, there is too much peptidoglycan in the gram-positive bacteria, and just because of that peptidoglycan layer, it can stand purple. And it is about the mechanism of the gram staining. Okay, so I have made a video over the mechanism of gram staining in more detail. So just watch that video just to make sure that just to clear up your this MCQs. Okay, so now the fifth one is that the genome of bacteria is normally in. Where does the genome of bacteria lies? So we all know that it lies in the nucleoid region. Okay, there mostly the nuclear material in the bacteria where it is present. It is that region is known as nucleoid because they are not having nucleus. Now, archaea bacteria and bacteria differ in there are two type of bacteria, archaea bacteria and eubacteria. So they differ in, if we look to the gene architecture, cell wall structure, gene transition machinery. So archaea bacteria are different from eubacteria with respect to gene architecture because the genes of bacteria and archaea are different. Cell wall structure is also different. Okay, and gene transition machinery is also different. Okay, the ribosomes are also different. So all of these. Okay, now 7.1, we can expect that one out of every bacteria will have a mutant characteristic. So it is not a technical MCQs, and the correct answer is 200. Okay, so we can expect that one out of every 200 bacteria we have a mutant characteristic. Now, the eighth one is that the genetic recombination occur in bacteria through the transfer of genetic recombination. Here come the genetics, so it is related to the nuclear material, and here genetic recombination can only occur through the transfer of plasmid. Okay, and we all know that what are plasmid? These are extra chromosomal nuclear content of the bacteria. Okay, so when these are transferred from between the bacteria, so there is a genetic recombination. Now chemolithoautotroph bacteria include which type of organism it include? First of all, you should know about the chemolithoautotroph. So chemo means that these are type of, these are nutritional type of bacteria. Okay, so and I have made a video over these 5 types of nutritional types: the chemolithoautotroph, the chemoorganoheterotroph. These are 5 classes of 5 classes, and how these are differentiated and how they get their name. So I have made a video over it. You can watch it in my videos. Okay, but here, which is the suitable answer? So just look here. Chemo means that it can get energy from inorganic molecules, and litho means that it can get its electron from inorganic molecule. Okay, and autotroph means that they are using CO2 as their carbon source. So just look here. If we look here to the halophiles, so halophiles are using salt. They are using salt as their electron source and is their energy source, but they cannot produce, they cannot use CO2 as their carbon source. So A is not correct. Okay, the second is myxobacteria. So myxobacteria are heterotrophic bacteria. Okay, and here is the autotroph, and spirochetes are also heterotrophic bacteria. The D number, the D is correct because nitrifiers are mostly it include the cyanobacteria. Okay, so these are autotrophic, so the D is correct. Now the 10th one is a bit out of course, and it is not included in the in this, so I have skipped this. Okay, just don't look to it. The 11th one is the three domain whose system of phylogeny incorporates standard molecular approaches. Okay, to classification but emphasize. So how, according to three domain system, the organisms are simplified or classified on the basis of ribosomal sequence. Okay, keep this thing in mind. The 12th is a method that some bacterial cells are undertake to survive adverse condition is So it is again repeated that some bacteria can undertake the survive adverse condition by the establishment of endospore. Okay, if there are certain adverse condition for bacteria, certain bacteria, so they are transformed into endospores. Now here is, if an F positive cell is put into a culture consisting only of F negative cells. Okay, so what will happen? First of all, you should know about the F positive and F negative cell. Now here is a bacteria. Okay, and here also it is a bacteria. So we all know that there is plasmid present in bacteria. So if I want to tell you guys that some bacteria does not contain plasmid. So if just look here, if this is a plasmid, so if it contains plasmid, so it will known as the F positive bacteria. And if it is not containing plasmid, so it is known as F negative bacteria. So first of all, I want to tell you guys that if a bacteria is not having a plasmid, so that bacteria should take a plasmid from its friend. Okay, through conjugation or transformation or different other things. So it will form a tube, and after that this plasmid will be replicated, and after that this plasmid will also get the plasmid from it. So after that it is having a plasmid, and this F negative cell will also have a plasmid. So now here this is also F positive, and this is also F positive. Okay, in some cases what happen, that the plasmid that is transferred from the original cell to the cell that is not having plasmid. So when it is this plasmid is transferred to the F negative cell, so a part of a plasmid incorporates into the nuclear material of the original nuclear material of the bacteria. For example, this is a bacteria. Okay, and it is having a plasmid, and here is a bacteria, and it is not having a plasmid, but and here is the nuclear material of that F negative F negative cell. So when this plasmid is transferred to this cell, to this bacteria, so the part of the plasmid or even all the plasmid is incorporated into the DNA of the that cell. Okay, the DNA of the dead cell. I'm just drawing the round shape, but it is not like that. So after that, this cell is now known as when it is having the plasmid that is incorporated into the DNA of that cell. So now that is known as the it is known as the HTR. Okay, it is known as the HTR plasmid. Okay, HTR cell. HTR cell. So this was all about it. Now just look here. If an F positive cell is put into a culture consisting only of F negative cell, so what will happen? Now just relate your answer to it. Okay, just look here to the C part. Almost all cell will become F positive. All cell will become F positive, with a few of them becoming HFR type. Okay, this is HF R type. Okay, it is not HTR, it is HFR type. So this is the correct answer. Mostly the bacteria will get the plasmid from its friend. It will mostly, they will become F positive, but some of the bacteria will become HFR type, because some of the plasmid will be incorporated into its natural DNA. Okay, so this is the MCQ. Now, if we proceed further, okay, the 14th number is, it is important to complete a course of antibiotic when they've been prescribed by a physician, because the correct answer is that if the antibiotic are prematurely stopped, some pathogens may remain alive due to their partial resistance to the antibiotic. Okay, so natural selection may reinforce this resistance. So the correct answer is B, because if a physician prescribed you an antibiotic for a certain period of time, so he knows that if someone stops the antibiotic in between them, so that bacteria can become resistant. So okay, so if you stop it in between the course, so they can become resistant. Okay, so this is, and the three options are not accurate. Okay, they are not relying. You look here, antibiotic are expensive, and these are not the conceptual MCQs about it, answers about it. Now the nitrogen in the atmosphere is made available to ecosystem to the action of the soil microbes. And what happens? The N2, okay, there is an N2 in the atmosphere. So what does bacteria do with this N2? It breaks the triple bond in the N2 gas and creating ammonia. Okay, it is nitrogen fixation. Now the microfossils about 3.5 billion years old have been found. They represent, what they represent, the remain of prokaryotic cell, the first organisms. Okay, I want to tell you guys that the microfoils, and just look here to the time, 3.5 billion years old have been found. So what will they represent? And we all know that when the world was formed, so there were only prokaryotes. Okay, there were only prokaryotic cells. Okay, so just because, just look here to the time, so the longer the time, the longer will be the most will be the remains of the prokaryotic cells, the first organism. Now the seventh one is the chromosome of bacteria are typically, so we all know that the chromosomes are bacteria are circular and double-stranded DNA. Okay, and it is found in the nucleoid region. Okay, horizontal gene transfer is a movement of gene from one species to another. In prokaryotes, this can most often occur. Yeah, so I want to tell you guys that horizontal gene transfer is also transformation, it is also transduction, and it is also conjugation. Okay, so here all of these cause horizontal gene transfer. Okay, so this is the MCQs about it. Now, the term autotroph refers specially to, the term autotroph specially refers to. So first of all, keep this thing in mind that autotroph means that it is using CO2 as their carbon source. So just look here, cell which can obtain their own food by engulfing other organisms. So it is not the correct answer, because it is not engulfing other food, it is producing itself. Okay. Now here is the organism which get their energy by harnessing the energy from spontaneous chemical reaction. So it is a bit true, but first we should know about the C option. Cells which use energy from light reaction but obtain carbon from organic molecules. So it is not correct, it is not obtaining carbon from organic molecule. The metabolism involves carbon, organic carbon and energy source. So here the organic carbon is not used. It can only use the CO2. So the correct option is the B. Okay, it is getting its energy from spontaneous chemical reaction. If I want to show you, so in the photosynthesis, what happened by the application of the sunlight, the electrons are energized, and after that they flow in the electron transport chain of the photosynthesis, and spontaneously it is producing energy. Okay, so that's why this is the correct answer. Now again, that this is repeated, so it is not included in it. Now an example of exception, use of structure of, here this MCQ is related to the this means that in certain cases the organelles that are used for a specific function in a cell, they can also perform different other function as well also. So it is known as exceptions. Okay, so and an example of exception is the transfer of toxins using a membrane protein complex and the dash from which it is originally evolved. So toxins can be transferred from one cell to another cell with the help of different other mechanism. But if they are not properly working, so flagellar collar is mainly used for the motility of the cell, but it can also transfer toxins in between the cells when it is required. Okay. The 22 is the in spore formation, the stage in which DNA condenses and align itself in the center of the cell is called So it is the first stage of the spore formation, and it is known as the mother cell. Okay, so the first stage is that the nuclear material will be condensed when the spore is going to form. The second is that the spore layer containing peptidoglycan layer is the cortex. Okay, the cortex mainly consists of the peptidoglycan layer. Okay, now if we proceed further, the 24th number. Okay, as calcium enters the spore, as calcium enters the spore, so water follows it into the when it is about the endospore. So when calcium entered the spore, so water is removed from the cell. Okay, keep this thing in mind. There is a very low content of water in the endospore. So when calcium enters the endospore, so water is removed from the endospore. Okay, the 25th is that once they evolve, they forever change the atmosphere on the earth. So we all know that when cyanobacteria was produced, so it changes the atmosphere of the world, means that it produces oxygen, and from 1% the oxygen level was increased to 21%. Okay, now the 26th is that which of the following statements are not true of about the prokaryotic cell. So prokaryotic cells are multicellular. No, prokaryotic cells do not have a nucleus. Yes, they are not having nucleus. Prokaryotic cell have a circular DNA. Yes, prokaryotic cell have flagella. Yes, so it is not true. Okay, they are not. It is true of prokaryotic cell do not have a nucleus. Okay, so which of the following statements is not true about prokaryotic cell? So the sorry, this one is this one. A number. A is the correct answer. That it is not. It is not. These are not multicellular. Okay. Now 27. When bacterial cell walls are covered with an outer membrane of lipopolysaccharide, they are gram negative. Okay. They are gram negative. Yes, lipopolysaccharides are only present on the gram negative bacteria. The prokaryotic genome is contained in the nucleoid region. Okay, as I have mentioned in the previous slide. Okay, prokaryotic organisms that obtain their energy by oxidizing inorganic substances are called. Okay, now it is now here it is again about the chemo autotroph. So prokaryotic cells obtain their energy, okay, the energy by oxidizing inorganic molecule. So when it is oxidizing inorganic molecule for energy, so these are known as chemo. Okay, so this photo and this photo will be removed. Now we will look for the chemo and chemo. So one is the chemo autotroph and one is the chemo heterotroph. Okay, so prokaryotic organism that obtain energy by oxidizing inorganic substances are called. So here is also auto and here is also hetero. So you will give preference to the auto because that it is oxidizing energy, it is using energy, it is using inorganic molecules as an energy source and as an electron source, and so the first is also inorganic, the second is also inorganic, so then it will use CO2 as their carbon source. Okay, so you should relate these three. So if two are the inorganic, so the third will be also the inorganic. Now what does it, so the correct answer is the chemo autotroph. What disease is experiencing new outbreaks because of antibiotic resistance? Okay, so here is the tuberculosis. Nowadays, just because of the resistance, antibiotic resistant, tuberculosis is very much evolving. The disease section referred to as silent sexual transmitted disease because it is usually asymptomatic in women. Early on is the, and here is the, it is not technical, it is now a straightforward MCQ. Here is the chlamydia. Chlamydia is a bacteria that is involved in the sexually transmitted disease. Here on gonorrhea, syphilis, these are also involved in sexually transmitted disease, but the diseases that are involved in the chlamydia are a bit asymptomatic. Okay, silent. Okay, so that's why it is chlamydia. Which of the following can be attributed to bacteria? Okay, so decomposing of dead organic matter, yes. Increasing oxygen level in the atmosphere, yes. And the production of antibiotic, yes. So all of are correct. Now the 33rd one is the how canners pressure cook vegetable as a precaution primarily against. Okay, and this is an MCQ that how canners Okay, how can a special cook vegetable as a precaution primary again? So we mostly cook our vegetables in the pressure cooker just to make sure that the endospores are killed, okay, and the bacteria that are producing endospores are killed. Now photoautotroph use, okay, photoautotroph use light as an energy source and can use water or hydrogen sulfide as a source of electron for producing organic compound, okay. So these are about the photosynthetic bacteria. Which of the following statement about the two domains of prokaryotic is not true, okay. So here is that the these A, B, C, if you read it out, so these are correct, and the third and the D number is the, it is false. And this is that of the two groups, eubacteria are more closely related to domain eukaryotes. So these are not related, okay. There is a very huge difference between prokaryote and eukaryote. Okay, a prokaryotic genome is different from a eukaryotic genome in is that just look here, that it has a less protein associated with its DNA and is not enclosed in a nuclear envelope. Okay, and the correct answer is C. A prokaryote genome is different from a eukaryotic genome. Is that it has less protein associated with its DNA. The prokaryotic DNA does not contain histone proteins, but the eukaryotic DNA contains the histone proteins. Okay, and it is also having the nuclear envelope, and eukaryote or prokaryote is not having nuclear envelope. So the correct answer is C. Okay, the first prokaryotes were probably cyanobacteria. Okay, sorry, it was not cyanobacteria. First, these were the chemoheterotroph. Okay, first these were the chemoheterotroph that used abiotically made organic compound. First, the prokaryotes were not able to produce oxygen. First, they were chemoheterotroph. They were using inorganic molecules and organic compounds for their food. And after that, then cyanobacteria was evolved. So the correct answer is B. It is a bit technical one. Okay, so keep this in mind. Banded iron formation in marine sediments indicate that, if I want to show you the banded iron sediments, and the last I will show you. So the correct answer is A, because that first of all, when cyanobacteria was produced, so they were producing oxygen, and that oxygen was reacting with the iron in the sand and in the rocks, and after that iron oxide was produced, that were producing bands in the rocks. Okay, I will show you at the last the bands. Okay, that what types of bands were they producing. If again in here I show you, these are the bands. Okay, these are the iron oxide bands, and these were just because of the cyanobacteria. Okay, now if we proceed further, here is the, which of the following statements about prokaryotic flagella is not true? So the flagella are composed of several microtubules, proteins surrounded by the plasma membrane. All bacteria has flagella. There is always only one flagella per cell. Motion is produced by rotation. So which of the following say about prokaryotic is true? So motion is produced by the rotation of semi-rigid helical filament. Okay, so this is the correct choice. Okay. Penicillin found as antibiotic mainly by inhibiting the ability of some bacteria through synthesizing normal cell wall. Okay, so penicillin inhibits cell wall formation, and how it inhibits cell wall formation. So I have made a video over it about the mechanism of penicillin. So watch that video. Plant-like photosynthesis that release oxygen occur in the cyanobacteria. Okay, dash are the surface appendages that allow a bacterium to stick to a surface. Okay, so these are the pili. Okay, pili allow the bacteria to stick to surfaces. What is the function of a bacterium's capsule? Okay, so the main function is the protection. Okay, it is also used in adhesion, but the most accurate function is the protection. Where is a bacterial cell DNA found? So it is found in the nucleoid region. In a bacterium, where are proteins synthesized? Okay, so in a bacterium, the proteins are synthesized mainly in the nucleoid region, or sorry, in the ribosome. Okay, ribosome synthesize the protein. What name is given to the rigid structure found outside the plasma membrane that surrounds us and support the bacterial cell? Okay, so this is mainly the cell wall. Okay, this is the cell wall. This is the rigid structure. Okay. Now the 47th is the dash is the bacterial structure that acts as a selective barrier. So plasma membrane acts as a selective barrier. Okay. The 48th number is that scientists hypothesize that the O157 H7 strain of E. coli is so different from the K12 strain. K12 strain because so I want to tell you guys that this strains develop just because it takes different genes from other bacteria. So E. coli takes different genes from other bacteria. So it is not just because of the horizontal gene transfer. So by horizontal gene transfer, this bacteria takes many genes from other bacteria and evolve itself to O57H7 strain of E. coli. So this is just because this. The second last is the prokaryotic cells that build stromatolites are classified as cyanobacteria. Okay, stromatolites are produced by cyanobacteria. The last one is that the prokaryotic cell that were the first to add significant quantities of oxygen to Earth atmosphere are classified as cyanobacteria. Okay, so these were the 50 MCQs. In the next video, I will discuss the 45 MCQs, the later 45 MCQs. Okay, so stay tuned and watch all the two parts. Okay, hope you this MCQs have helped you a lot. Okay, so check yourself that how much of you have corrected it. Okay, now thank you very much. God bless you.